Sure.
Voila.
Okay. Yeah, I will pause it.
Year 2023, which was an amazing year. We really published a lot of really good stuff on the Stockholm Paradigm and DAMA and host parasites and parasitology. And the journal has really taken off, and I really appreciate everyone's contribution to that. So today we will have Valeria Trevoloni, and I'm going to turn over the introduction of Valeria to Dan. And so the interesting thing here is that I'm running this talk from Montgomery, Alabama. Dan is in Asheville, and Valeria is over there in Illinois. So it's an amazing group of dispersed people, but that's how I think is really making it exciting now. So go ahead, Dan, take over the introduction, and then Valeria, then you can go forward, and we have the recording started. So thanks a lot, everyone, for being here. I appreciate everyone's participation.
All righty. Well, it is my pleasure to introduce today's speaker. Valeria Trevoloni is an ecologist and evolutionary biologist whose work integrates ecological theory of community assemblage and coexistence within an evolutionary perspective to explain the origin and evolution of host pathogen systems, both in the wildlands and in the agroscape. She's currently an assistant research scientist in vector ecology at the University of Illinois, Urbana-Champaign. She earned her master's degree in environmental sciences from the University of Pisa and her PhD in biology from the University of Neuchâtel. In her thesis work, and as a research assistant following that at the Swiss Institute for Forest, Snow, and Landscape Research, she developed a conceptual framework for selecting suitable bioindicators of high levels of taxonomic and functional biodiversity in agroecosystems. In 2008, she moved to the US, working as a postdoctoral fellow at the Illinois Natural History Survey at the University of Illinois. During that time, she created the global Hemiptera phytoplasma plant biological interaction web interface, created and published an R package to simulate the dynamics of host switching, and contributed to further development of the Stockholm Paradigm and DAMA. With her research program focusing on re-emerging infectious diseases and food security, she is uncovering new plant pathogens in natural habitats and filling knowledge gaps for a group of bacteria called phytoplasmas. Her research interests range from understanding the relationship between global changes, such as land use and climate change, and emerging phytoplasma diseases, leveraging data from archives and collections to discover new strains and hosts of phytoplasmas, and use of georeferenced satellite images and land use regression models to assess the risk of emerging disease. She's published more than 130 articles in refereed journals and 6 book chapters in her short career. And it is a great pleasure for me to call her a young friend and to introduce her to you.
Thank you. Thank you very much, Dan, for this introduction. And thank you, Scott, for inviting me to this seminar series. So today I'm going to give some information about emerging vector-borne diseases, plant diseases specifically, and how to apply the DAMA protocol using this amazing study model that I've been studying for a couple of decades. And also I'm talking about One Health, but this is just a corollary. So first of all, acknowledgements. I really feel very lucky to collaborate with this amazing group of brilliant researchers. So I really want to thank Dan, Eric, Walter, Scott, Sabrina, Orshi, and all these people that gravitate around the Stockholm Paradigm group, because they give me a great opportunity to integrate plant diseases into this big dialogue of biosphere, evolution, emerging infection diseases, and is a very nice collaboration as well. And then, of course, I would like to thank you, Chris and Brenda from the University of Illinois, for contributing significantly to my scientific growth. So this is first. And then we can talk about the research. So this is the outline for today. So I'm talking about main gaps and misconceptions around the One Health initiative to cope with emerging infection diseases, and then about the conceptual proposal of the Stockholm Paradigm, and then I will focus on vector-borne plant diseases, and then I will showcase how to cope with emerging vector-borne diseases using the DAMA protocol. So let's start from this. So One Health is a simple and powerful concept to cope with emerging infection diseases because wants to build a consensus upon unified solutions to protect and guarantee health for everyone and everything. So, however, it was considered conceptually and operationally incomplete and is broadly characterized by a response-based approach. So let's start from the definition of One Health, which is slightly evolved over the past 14 years. But in 2022, One Health high-level expert panel provided this working solution that want to integrate principle embraced by several other initiatives such as eco-health and planetary health. So this seems the most holistic definition we have. However, I want you to look at the highlighted test carefully. So we have One Health is an integrated, unified approach that aims to sustainably balance and optimize the health of people, animals, and ecosystems. And it recognizes the health of all these things is linked, interdependent, and blah, blah, blah. And then we have eventually plants and environment, including ecosystems and blah, blah, blah. So what I think here is that this wording is somehow misleading. And this is because of the epistemic gap. And I'm being fair because sometimes I would like to evoke the epistemology of ignorance. So this epistemic gap here is the lack of understanding of the existing distance between how we perceive and manage the ecosphere and what we really need to achieve the noble goal of One Health. So because of it, we have limited possibility for becoming critically aware of this situation. And we know that to fill this gap, we need critical learning processes, such as neutralizing this phenomena, such as the epistemology of ignorance. So we need to reevaluate old paradigms. We need to contextualize didactics, and we need absolutely an operational workbench. So the greater the gap, the greater is the defensive tendency to believe that health crises are fortuitous and unpredictable. So let's understand this epistemic gap using the diagram provided from One Health. Let me take the pointer here. So we have this broadly accepted one, which is a triad in which we have animals and then we have humans, the most important species on Earth because they serve an entire sphere, and then the environment upon which animals rely. And we perceive this sphere as everything else, non-living things, plants, etc. And this is clearly an anthropocentric and zoocentric view. At the same time, because it was recognized that plant health and related diseases were not considered or integrated into the One Health concepts, some authors in 2022 proposed a quite complicated stack of two nested tetrahedrons, which distinguish human being again from animals. But we have the plant sphere here. After all, we also have another sphere, which is the pathogen as a separate entity. And this makes the overall bias even worse for microorganisms in general and for pathogens that are depicted here as the bad guys. But sometimes they are not. So interestingly, these authors also include the temporal dimension, representing this evolution of the disease. So to fill the epistemic gap, first of all, what I'm proposing here, we need to equally include the components of the ecosphere and leave the temporal component for the next step of conceptualization and implementation. Secondly, we need to understand that the only thing that distinguishes humans from all the other things is their consciousness, which open another philosophical dialogue about stewardship and responsibility. So at the end, we have one sphere for all the animal, the plant, the abiotic and microbiological sphere, and all of these spheres may be affected by diseases or other kind of stresses. So the question now is, how do we know that the One Health initiative are not working? So in 2019, the World Health Organization published the top 10 public health challenges and responded with a 5-year strategic plan, mainly based on preparedness, prevention, and surveillance. And these are the emergencies listed. In 2022 and 2023, an update was published. You might want to see here the citation. And the others on the top of this 10 included more emergencies for the global health, like monkeypox, conflicts, reproductive health, mental health, and so on. And in 2023, the WHO reported some more, such as cholera, RSV, Nipah virus, and so on, and then launched an appeal for $2.5 billion more to cope with these health emergencies. So now you have noticed that about half of them are infection diseases, and all of them are zoonotic diseases or diseases associated with humans. And plant diseases rarely, if at all, enter in this big conversation that regards global health. So clearly, we are missing the opportunity to properly handle food security and global health. So when we talk about emerging infection diseases crisis, we actually think about zoonotic diseases. So this is a picture by Fauci in 2017 that summarized the spike of new emerging infection diseases events in the last 2 decades. And this is roughly correspond to when the One Health initiative was established. So One Health initiative is struggling with zoonotic diseases. And moreover, we also know that similar global overview for plant pathogens are not available, and only a few of them were actively monitored globally using this advanced surveillance systems. So here, for example, on the top, emerging fungal plant diseases that increased by 5.4% from 1994 to 2010. And on the bottom, another example about the global trend of the dispersal for the cucumber green mottle mosaic virus since its first emergence in England in 1935. So in 2021, the Consultative Group of International Agricultural Research, which is this global partnership for food security, organized this webinar series, and this was a very good opportunity because they finally recognized that plant health is a key for One Health approach. And this is very important because we need to integrate One Health to achieve food security, zero hunger, to reduce poverty by ensuring access to nutritious and safe food, and blah, blah, blah. But in terms of initiatives, as you can see here, a couple of examples, a lot was done for livestock diseases and for foodborne diseases, and you can search online. There are a lot of details and initiative about that, but little progress has been made for plant diseases. So we have several interesting review papers on this topic, so how plant diseases are linked to food security. And in one of our reviews, we summarized direct and indirect economic impacts due to most important emerging plant and livestock diseases here. So this is just a snapshot. Don't want to be comprehensive, but just want to show you that the burden of emerging plant diseases is recurrent, is geographically well distributed among food secure and food insecure countries and is unsustainable. In this other paper, we presented the graph of the cost of the new emerging diseases here, that after the first 20 years is internalized as a fixed cost that became cumulative. And on the bottom, we have the trend for the next 70 years, where the cumulative costs of emerging infection diseases in crop and livestock in the United States will increase exponentially, ultimately exceeding the curve of the predicted GDP. So, because plant health is key for food security and One Health, here I'm proposing to formalize the relationship between plant diseases, which is considered independent variable, and human health, which is considered the dependent variable, including the mediated effect of food security. So this is a simple mediation model. And in this model, plant diseases is expected to increase food insecurity, which in turn affect human health. So overall, plant diseases affect human health through a direct impact and an indirect effect mediated by food security. So this is what the One Health high-level expert panel published in 2022. So you can go and check on the paper, it's pretty short. They recognize that because One Health must deal with urgent global issues, they want to translate the concept into policy and concrete action. But the point is, this is really difficult if we start from floated conceptual basis and incomplete operational proposal. So again, the broad approach of the Stockholm Paradigm would be a good opportunity for the One Health initiative, and in particular for the to cope with emerging infection diseases and to fill the gaps that I presented yearly. The Stockholm Paradigm has been presented in detail by several people during this seminar series, Salo, Scott, Walter. So I'm not going to go into each single detail because they already did an amazing job to do this. So I'm just going to summarize here a few concepts that will be functional for this talk. So we are losing the battle with emerging infection diseases because all the initiatives are based on a traditional theoretical framework and misconception about nature and evolution. Moreover, we have this traditional evolutionary view that wants that parasites and hosts to be associated through a strong co-evolutionary relationship. And this view generates what we call the parasite paradox, which means each pathogens tend to be strictly associated with a single host species, so that the likelihood to colonize different species is very low. And this would bring to someone knocking my door, I don't know, sorry about that. We should, in this view, we should not observe an increase of emerging infection diseases event, but this is not the case, obviously. So the Stockholm Paradigm basically integrates Darwin's fundamental vision of the nature and want to resolve the parasite paradox, relying on the notion that emerging infection diseases are expressions of pre-existing capacity of pathogen that colonize susceptible but previously unexposed hosts. And finally, the DAMA protocol is its operational proposal in the context of emerging infection diseases. So now I'm going to focus on emerging vector-borne diseases, because we all know that vector-borne diseases pose a major threat to public health and agricultural system globally. And of course, we have several results and initiatives for vector-borne animal diseases. So I listed here some examples and results and initiative from WHO. But I want to point out that the Institute of Medicine, that now is the National Academy of Medicine, published a workshop summary in 2008, and later another in 2016. Those are pretty interesting, on global health impacts of vector-borne diseases, where they advocated a better connection with One Health. But at the same time, they still observe a lack of the interconnection, even between plant and animal vector-borne diseases approaches. And they also notice a specialized tendency to each single approach and this technology-centric view. So a lot of gaps, a lot of missed opportunity. However, we all agree that vector-borne pathogen in general have a complex transmission cycle. They also have this high turnover in many different alternative hosts. So usually the vector host pathogen system is represented as these three entities that interact with one another. So we have the group of hosts, two in here, to which we give different qualities, right? So we can have like primary host, alternative, reservoir, final host, susceptible, not susceptible, depending on which angle you look at the model. And then we have the vectors, and in here, so mainly are arthropods, but there are other representative. And I'm leaving out the fomites here because we are not talking about that today. And then we have the pathogens. So it was noted before that vector-borne pathogen that there's multiple hosts require multitrophic models to evaluate circulation and disease dynamic, but the system is very complex. So my research focuses on a group of plant pathogen that provide a very good example of the complexity of the interaction. So the plant here is considered the final or primary host, and then we have the Mithra insects, which is a group of biting phytophagous arthropods, and of course they are the vectors, and then we have a Phytoplasmas, which is a group of bacteria in the class Mollicutes. So let's have a look into Phytoplasmas a little more in detail. And I like to provide few information using, on the left, we have this tree of life, a good one, where we have Phytoplasmas in here, in this clade, which is the phylum of Mycoplasmatota, formerly called Tenericutes. And if we zoom into this clade on the right, we provided the time tree for Phytoplasma, including the main representative in the class of Mollicutes. And then we have the order Acoleplasmatales, which is green plus this clade in blue. So of course, we have all the reference strains of Phytoplasma, which is this monophyletic group here. So a few facts about Phytoplasmas: they are strictly transmitted by Hemiptera insects. They live exclusively in the plant phloem and insect tissues, and then they are able to elude host defense and exploit metabolic pathways. They, of course, may cause severe plant diseases. So we can talk about plant diseases, economic importance for hours, but what I want to do here today, just to report a recent strategic plan that was published this year by Harper and collaborator at Washington State University. And this is for stone fruits, which is a multi-billion-dollar industry in the United States and Canada that suffers significantly from these economic losses due to outbreaks of X disease Phytoplasmas. And the most recent outbreak in the US Pacific Northwest resulted in an estimated like about $65 million distributed from 2050 to 2020. And here you can see in the US Pacific Northwest, the emergence and reemergence of this disease during this period of time. So now let's go into some detail of the transmission mode. I'm not going to be specific, but I prepare an animated cartoon for you to understand that these Emittera insects feed on the phloem of an infected plant, acquire phytoplasma, which is this cell moving into the midgut, and in the midgut interact with the membrane protein, migrating into the hemocoil, and then start reproducing, colonize the salivary glands, and then go into the saliva, and then can be injected into another plant. And into the plants, in the phloem, the phytoplasma is able to active reproduction and this dispersal across the seeds. So this mode of transmission in the insects is called persistent propagative. So now, from the perspective of the trophic interaction, this is how we used to break down the complexity of this relationship, right? So we basically have three bipartite relationships: plant insects, plant phytoplasma, and insect phytoplasma. And there is this color code, with blue being the pathogen or the commensal. It depends. And in black, we have the host that can be, sometimes can be the insects or sometimes the plant. So, but when we look at this relationship from this evolutionary point of view, we know that there are different interpretations on how each bipartite relationship established and species evolved. And in particular, plant health professionals and different groups of researchers used to apply this traditional view that is characterized by at least one of these three aspects that I'm going to list. So the first one is focusing only on the host-parasite relationship that involve the species we care about. So, for example, in this case, the diseased crop. The second point, that's many times they focus on a few single vector species, which are always the most abundant or those that are easy to identify. And lastly, and more importantly, the observed relationship is interpreted as locked into a strong coevolutionary dynamic that want the parasite to be like this in constant coevolutionary arm race with their hosts, which under natural selection eventually leads to increased specialization. And you can see this perfect coevolutionary relationship depicted here. So by reviewing this tendency to interpret the association as such, I discovered that for plant diseases, de Bary was the first setting the stage for fungi establishing this one-to-one association with the host. So he called that le choix rigoureux, which just means you don't have any other choice. It's just one-to-one, which also was used to establish parasite species name. So several authors followed the system of naming parasites after their association with a specific host. So from an evolutionary perspective, this was translated in what we call today the Dietel's laws in 1904. So basically, this law wants that this strict coevolution among parasites and hosts, and several other, of course, embrace this paradigm. However, we published recently a meta-analysis of all the studies that formally tested for cophylogeny, which is interpreted as coevolution most of the time between interacting species. And it turns out that for parasitic association, as you may want to see here, so the graph depicts the predicted probability of a congruent outcome. And you can see here that for parasitic, using the event-based approach, which is one of the most reliable, but I'm not going to talk about that in detail. So we can see that there is a clear tendency that the congruence outcomes are quite low overall. So unfortunately, a few papers that I reviewed for this meta-analysis included the vector-borne pathogens. Many of them were just review papers, not including any statistical, formally statistical analysis. So I just enjoy reading course, and I will provide you with an example of one of them. So this paper was published. This is pretty interesting paper if you are interested in vector-borne plant diseases. It was published in 2011. And this is clearly show you the traditional view. And this view emerged from few facts and sentences here and there. So first of all, they say that phytopathogens have evolved specialized traits that enable them to colonize specific plant hosts and cause disease. So the view wants that you first evolve something and then you are able to colonize. Otherwise, you cannot. And then sometimes, for this specific situation, they evolve an alternative association with herbivorous insects. So now, when the things became complicated for them, because they recognize that there is an emerging paradigm that wants insect vectors serve as alternative primary host. And they create this dualism being a vector, or rather being a host, or being both, based on these trade-offs between host symptoms and pathogen fitness. So in this view, if a vector is a vector, then is a generalist. If the vector is a host for the pathogen, then the pathogen is a specialist, and the association go towards a specialization. And they concluded that if the vector is a vector, then there's one insect for all occasions, they say, I should quote it. And if the insect is a host, or as they say, alternative host, the parasites already evolved specialized molecular traits in order for it to be specialized on the insects. And then when the vector is both, this is just may represent a rare transitionary stage in this evolution of insect pathogen association. So this is pretty complicated from my point of view, but again, from this background, we have the parasite paradox. So from one end, parasites goes toward this extreme specialization with this final host, which is the plant. But from the other end, with vector playing the role of carrier vector, it is not clear how pathogens can cope with new opportunity to change plant host or even to change vector. So finally, if the vector is a host, again, the pathogen is evolving specialization. So in any case, we are going to face this paradox. So the alternative view of Stockholm Paradigm, again, that start from this fundamental observation by Darwin, in this case, may be applied easily to each bipartite association. So we may say that the nature of the organism, the pathogen, is more important than the nature of the condition, the host. And the pathogen are characterized by the inheritance that is highly conservative, produce variants, and is historical. And then pathogen can colonize new hosts through this accumulated capacity that they already have. And there will be a certain misfit with the new host. And then this is when finally acts the natural selection. So in the vector-borne system, all the steps are applied in each of these bipartite. And also we know that emerging vector-borne diseases occur when pathogen interact with another host through host switching. So this is a lot of words, and this has been presented here earlier by other very well. So I like always to depict with a cartoon this idea, and here to represent the host switching and the potential for emerging diseases, I'm presenting a cartoon that basically depicts the original idea formulated by Augusta and Clemens in 2008. This idea was further modified, the graphic and the overall interpretation by Orshi during the seminar given a few months ago. And here I'm slightly modify further the cartoon to accommodate the vector-borne pathogen system, but basically is the same thing. So we have first this ancestral association, and what it is basically is the pathogen and the vector. So the vector is represented by the axes that are the operational variables, which has this variation in here and in here. And this gray oval here is the pathogen that is living on this host. But then in the same way, we have the association between pathogen and plant. So we have another diagram in here with operational variable 1 and 2, which characterize the plant. And in the same way, the pathogen here that is living with its host and have been accumulating a certain genetic variability. So the dimension of the oval is just the genetic variability of the pathogen. So now what we have I added more in here is that depending on the variables defined by the tree-trophic interaction, so an infected vector interacting with an healthy plant, or a healthy vector interacting with an infected plant, all of this constrained in the ancestral component of the healthy vector, healthy plant relationship, we have immediately this third dimension with all these slices of possibility driven by the multitrophic model. So this three-dimensional space include all this possibility in time and space. So now, during a colonization event or dispersal event, for example, let's imagine that this pathogen, this same pathogen, colonized another vector species with, you can see here, different variables on the axis, but keep the same ancestral plant host, or even colonize another different plant host which has this different variable here. So what happened overall in both cases, we have a subset of the population of the pathogen that can still colonize, be able to colonize the new host here and here. And actually it's not a fit, but it would be a misfit, but overall we have a successful colonization, and over a reasonable amount of time, this little portion of individual will start to reproduce, diversify, accumulate genetic diversity, ultimately exploiting the new hosts and generating new spaces, new three-dimensional spaces that I know is difficult to imagine, but this is the idea basically, right? So all this reasoning over the tree-trophic association between phytoplasma plant and vector allow us to better interpret all the patterns that we also observed using different analytical approaches. So, for example, here in this paper that we published in 2020, we used a time-tree reconstruction for phytoplasma and to infer and to get some indication for ancient expansion event on different hosts by phytoplasmas and subsequent diversification events. So, for example, here in this point, phytoplasma possibly colonized Hemiptera insects when they emerged, host switching from a possible vertebrate ancestral host, or maybe phytoplasma were free living and just colonized this new host, and then they also colonize spermatophyte, and then there is this diversification event, and later they colonize angiosperm, and then after this event, we have this huge radiation of different clades of phytoplasmas. So then we hypothesized that vector competence was phylogenetically conserved in specific clade of Hemiptera and associated with specific traits of phytoplasma that are phylogenetically conserved. So this is the phylogeny of Hemiptera provided by other authors, and only few clades retains competence to transmit phytoplasma. These two in yellows, this and this. And vector competence seems associated with anomoplastic traits. But if we consider this group here, which is the big group of Membrachoidea actually include the higher number of vector species that transmit phytoplasmas. So what we did, we chose this clade, we selected 3 representative species in this big clade, and we used a protein-protein interaction experiment to provide evidence for vector capacities and traits associated to the capacity to be phylogenetic conservative. And basically, we provide evidence for this ecological and molecular misfit of the pathogen varying along a continuum in this among this related and unrelated species in the clade of Membrachoidea. So more evidence of host switching. We used a classical global fit cophylogenetic approach, and we provided evidence for a good amount of host switching going on. We have the blue indicating strict coevolution, the red indicating incongruence, basically. So not co-speciation. And the shade of the color is the strength, the significance of the results. So we got this good amount of host switching. We may infer different event of host switching from here. And we also know that there is only one group that provide the major evidence for coevolution. And this is this group here, which include Psyllidae in the Cacopsylla genus, and this group, monophyletic group of group 5 in phytoplasmas. This also suggests, host switching is also suggested by this ancestral state reconstruction made on the phytoplasma phylogenetic tree. And you can see from this ancestral host, which is in the tribe of Cini, there were several opportunities to phytoplasma to colonize different hosts, and sometimes even unrelated, distantly related, like in here. Like this is like clearly changing the rule of the game, and we have this distantly related colonization, and then several events of switching going on. So given all of this knowledge now that we have been accumulating, we take them into this operational protocol, which is linked obviously to all these concepts provided by the Stockholm Paradigm. So this is the DAMA protocol, and this is the framework that we have been using since several years now. So maybe this framework may be easily integrated to the One Health initiative because it's really operational. So what I'm doing here, I'm explaining in detail how this can be. So first of all, we need to recognize that we live in this complex and hyper-connected ecosphere. And when we are planning something that have to be applied, we need to consider this spatial scale and all the components of the landscape that interact. And in this context, other collaborator, so we in this group, we already contextualized the risk of emerging infection diseases in this landscape level. And we know that this risk increases along the interfaces among different habitats, and that the dispersal of the pathogen and the host switching is clearly triggered by stresses such as climate change, but even other stressors like land use change, globalization, trade, and so on. And of course, we know that this risk space is in continuous evolution. So the first step for the DAMA protocol is to document. So in this paper, Ober and collaborators pointed out the central importance of archives of information and physical specimens. So all these things like taxonomic inventories, bioinventories, any kind of database, all this data may be aggregated to be used to run several types of data analysis. But what is important here, that everything has to be linked somehow to vouchers, reliable archives, and so on. And then that, of course, will facilitate tremendously the assessment in the step 2 of the DAM, right? So here an example on how we document using biorepository. This is our current NSF project where we are screening potential vectors preserved in our biorepository at University of Illinois. And until now, we discovered 30 phytoplasma strains associated with previously unknown vector. You can see in red across this huge phylogeny of phytoplasma, all the discoveries that we made. And 11 of them are new group of subgroup of phytoplasma, not yet described. All this discovery were made all over the world because the collection include a lot of species collected everywhere, basically. And we have this very good geographic coverage, and this is what we got. So basically, with the second step of DAMA, what we do is to assess the risk of emerging infection diseases. So we could run different type of analysis, right? But at the end, what we need to do is to summarize all the data, all the analysis, in a way that they are readily usable by those that allocate funding for the monitoring, for the action, and everything else. So a very understandable and useful number or data for this stakeholder that can take decision easily. So this heat map here defines the level of risk for the outbreak. And the concept of the heat map here is really powerful because it's easy to comprehend and is also immediately immediately useful for the practice, right? So what the risk is the product of the likelihood and the impact of an outbreak. So we have this different level of risk from very low to very high and blah, blah. So what we have to do with DAMA is to take this heat map and build the risk upon the diversity, the phylogeny, and the historical ecology of the association that are the bipartite association, each of the three bipartite I showed you before, but ultimately we want to know about the tripartite association. So what happened when all these three things occupy a certain point in time all together, and what happened if this encounter is all red? So what happened if what we have to do if this encounter poses the highest risk? So eventually, the DAMA protocol has been showcased using data collected during several bioinventory projects. These projects were carried out in South Germany, this region that is called Bavaria. So I had this great opportunity to look into this data, and finally we published this paper and we presented the results that show basically how to produce actionable information for stakeholder applying the DAMA protocol. So this is the step of the DAMA protocol, how those steps were formalized for this case from Germany. So what we know is that having this huge amount of data available, what we can do now is to carry it out the phylogenetic triage here, which is a tool for the assessment. And this is a very easy approach because first of all, you have to answer some question that regards the specific characteristics of the tree, of the pathogen, the vectors, and the hosts that you have in a specific landscape or region or whichever spatial level you want to work. And after you answer this simple question, you need to evaluate the phylogeny, and then finally you can assign a risk to each association. And in this case, when plotted as such, each quadrant defines the risk for each tripartite association. The final goal of this paper was to propose a list of 6 vectors, and the 6 vectors come out from where the species encounter the susceptible host, the host that we care about maybe, or where this encounter happened when the pathogen or related strains are associated with this species. And this list is very useful for the monitoring point of view, because when we want to run a monitoring plan, we don't want to really monitor everything, but we want to target specific things. So we want to save money in the meanwhile and be the most effective as possible. So what we are proposing here is just 6 species of vector that more likely will pose the highest risk for the spreading of this clade, this group of related pathogen. So this is the conclusion. So my final remarks here, some steps for the future. So there is a lot to do, right? But certainly what I would like to do is to build an operative modeling platform to be implemented on DAMA protocol. So we have our colleagues, Walter and Sabrina, already working a lot on this point. They provide a very good modeling platform. I would love to see those really operative so that people can use it easily. And another thing that I would like to do with my colleagues is to integrate this Vector-Borne system into the individual-based model that Sabrina presented. So we are collaborating on it. We are talking about different aspects that unfortunately, for the sake of time, I didn't touch today, like the OS manipulation aspect and the paradox that arise from that one. I didn't have time to cover in this presentation, but this is certainly something that we are doing, and we will go in the next future. And then, as a researcher, another bottleneck for the full application of the DAMA is this early step, at least for me, is this early step of the monitoring. So we want our data to be used. So we produced actionable data. We translate basic research for the apply, to be applied readily. But now how this can go on, how we can implement for real, we can take action based on this data. So we have good example for successful application of the DAMA, and those are provided by this tick-borne disease study case in Hungary from Gabor Fumbari. So this is great. So we have to learn from that, of course, but we also need to implement better all this conceptualization for the policy and the action provided by Ocean collaborator. So this is what is in the future. And I think with this, I'm concluding the talk. Thank you.
Thank you, Valeria. It's a really nice talk. And I would like to open the floor for questions. And most of you, we have 25 online participants, and there are 4 of us here. So raise your hand and I will try to let you know who can talk first. Or so any questions? So Walter is saying, unfortunately, I will have to leave earlier. And if you were not here, Scott had to leave, so I took over as hosting this talk. And Scott might be already on an airplane coming back to Lincoln. So Walter is leaving, and John Genevieve, well done, thanks. And Walter left. So any questions? There is one, Sabrina. So I will hand over to Sabrina.
Hello, thank you. Hello, everyone. Hello, Valeria. Thank you for your talk. Nice talk. The last part, when you applied DAMA, you considered the tree trophic system. Yeah, you consider. Did you compare how your predictions would be different if you consider just plant and host? How your predictions change and we do just this traditional bipartite approach?
Unfortunately, this is the point that we tend to simplify the nature, right? This is a complex system. We need to simplify sometimes, even to understand the things. So even if we tend to understand the things like bipartite, then everything happen in the tripartite, three-dimensional space. So this is a fact. So it's kind of meaningless in this case to try to interpret just from the perspective. I mean, it's a very nice academic exercise, I think, but it's meaningless from the point of view of practical application, to say the least. But even from the academic point of view, because we know that until now, phytoplasma are strictly transmitted by vectors until now. I don't know if it was like this before, but for now, it's like this. So there is no way that even a population of phytoplasma may persist for longer just in one plant, even if you talk about individual plants. So you may want to consider anastomosis, blah, 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 but there is no way that phytoplasma in one plant may persist individually for a long time. So again, I cannot imagine even to
Yeah, no, I'm saying just not that it would happen, just how approach more simpler, more simple, you know, if you just to see how if you simplify even more, if your prediction change a lot. I'm probably, you know, that I'm asking that in the view of the model that use even simplify more than this 3-dimension, and to measure how important it is to consider the tripartite model.
In terms of the model, Sabrina, I think that we can simplify as much as you want. This is why I started, and I can continue to start from this bipartite. But at the end, we have to find a way for these three things to interact. I don't know if it's a product, I don't know which kind of mathematical strategy, but see what I mean? At the end of the day, we have to let them play together with all the probability associated to it.
Thank you.
Actually, I have a comment, or maybe it's a question. So I'm sitting across from the botany collection here, and my understanding is that the unique feature of these dried plants that you can extract perfect DNA from even 100, 150, 200-year-old specimens. Have you worked with these botany collections?
Yeah.
Yeah,
this is a really good question, because I have a collaboration with the University of Michigan. There is a very good collection there. And there is this very smart young curator, she contacted me and she told me, look, I have a collection of Prunus species from 1888. And the guy that collected was this agricultural driven person that left a lot of notes about being impressed by the symptoms and blah, blah. So she digged into the history. She discovered that these plants were all infected by phytoplasma. At that time, they believed that was a virus, actually it was phytoplasma. But anyway, what we are doing now, she's sending me all this old material. We created like a kind of experiments, like we created a time series, like trying to use all this advanced molecular media to extract ancient DNA. This is for the disease that I talk about today, the X disease that is affecting badly US and Canada. And this would be an ancient infected voucher. So I'm excited about it. And I'm excited to explore even further all the possibility to dig into this voucher that we have then being used for several other purposes, even for the perspective of emerging diseases. So yes, this is a very good point. And I will tell you something more. We have to find a way to dig into these vouchers in a non-destructive way. So there are a lot of possibility to do this, including imaging. So there is a lot going on.
Yeah, just commenting on the imaging, NSF was funding, it's called thematic collections, and they have these conveyor belts where they were imaging botany sheets one after the other, and in theory you can screen them just by the images, whether you see pathology of phytoplasma. So
yeah, exactly. Yeah.
So you don't have to dig them out or go to the very specific collection. You can pre-screen them based on images, and then request just maybe subsampling.
Yeah, and if we want to talk about alive species and we want to touch the issues with biodiversity and conservation concern, we can also use imaging on in vivo specimens to screen for pathogen when we don't want to kill everything, right? So this is another approach that I'm exploring currently here. Thank you for this question.
Okay, Pedro online is, I will ask him to unmute.
Hello. Wait, let me just turn on my camera here.
Oh, hi.
So, hi. Congratulations for the presentation, and thank you. It was very nice and very informative. I was just thinking, it kind of question that relates with the question of Sabrina. Like, how much could we? I understand that we have to simplify the systems, and I think it's obviously the feasible manner, to do it. But like, how much our knowledge about the broader network of ecological interactions in each environment that we are accessing could like enhance our predictability or the accuracy of the DAMA protocol and the risks of them.
So actually, I didn't have time to present here all of this. I did for other seminars, but our colleagues, especially colleagues that work in applied research, and they have been accumulating for years a lot of useful ecological information about each of these bipartite, right? So for this model is really, we are really lucky because we have a lot of data. It's just a matter to use the data into a proper modeling platform and into the DAMA protocol. This is, so the last showcasing that I presented today, and I've been presenting this year in several occasions is just because the data were available for other purposes and so on. But when you take the data all together, they are extremely powerful. And we have a lot, really.
Yeah, because that's nice, because I was thinking like how much like the risk assessment would change from like you have the same focal system, but in different environments with different diversities and different dominances of species.
I understand your question. Eventually, we work with Germany, so they spend a lot of time, money to collect several types of data. So we have the highest concentration of information there. Probably there are other regions where there is very much more of the unknown, but from my point of view, because this phylogenetically conservatisms across the phylogeny, right? You can build bigger phylogeny and you can at least infer certain traits for clades that even if are not distributed there, may behave the same way in another region. So the powerful of this framework that is based on this conceptual Stockholm Paradigm approach is that there is this huge data that come from the phylogeny that have to be integrated. So eventually, this may solve what you are asking about, the tracking of information.
Okay, thank you. Yeah, makes sense. Thank you.
Okay. I have one last question here from Ethan.
Hi. So you've been discussing, as with this previous question early in the slides, the need for a really good platform to apply the model. And I was curious. I know you'd alluded to work being done on it. Is what's missing like an API to make the various data formats from this data out in the wild compatible, or is it more of a user interface? Like what are the technical challenges, I guess, from like a end user, or in terms of like creating something that is user friendly and easy? I'm just kind of curious as to what the challenge is.
I'm not sure about technical challenges. I'm not an expert on this perspective, but what I was talking about was more like really what we are missing is really this tripartite component of the vector-borne pathogen that we need to implement. And I was thinking easier for me to start with the individual based model that Sabrina and Walter have been developing during these years. So for me, I already worked with them, so it's natural that I want to start from them. And then in terms of making this more operational, I don't know how we can do this, but eventually this will be necessary at a certain moment. We have to deal with it.
Okay. Cool.
Okay, I think I will close this meeting. Thank you, Valeria, for presenting on the very last seminar of this semester. This is just a reminder to the other participants that the semester ends this week. Is that right? Or
next week is
next week.
Yeah.
Finals week is next week for the students, and we will have the winter break. So I wanted to wish happy holidays to all the participants, and hopefully we will see you again. Scott will send out an email, so he already made some plans with Dan Brooks and himself to start another seminar series in the spring semester of 2024. So thank you again. And if
Thank
you have
you.
any comments or anything to say, this is the last call, but otherwise, goodbye.
Thank you very much.
Thank you.
Congratulations, Valeria, from Bruno.
Thank you, Bruno.
Thanks.
Bye.
Bye.
Albert, you're